See some dumbbells in the middle, which are actually disulfide bonds, two sulfur molecules binding to each other. They sometimes bind across proteins and sometimes bind within the protein. These create another level of rigidity in the whole molecule. The bottom line is, you just don't start with the chemical structure, but there's a whole lot of post-creation modification that has to go on before these things work. So what's the problem? The problem is you have a biologic drug, and the government wants essentially generics to come in. But the generic of a biologic is not necessarily a generic, the same as a generic of a small molecule. But with Hatch-Waxman in their brains, they came up with this scheme. As I mentioned, after the protein is produced, it still has to be modified. And that modification essentially has to be done in a living cell. So a biologic has to be created in a living cell. And not just any living cell. Different living cells will give the biologic a different structure. So it eventually has to be a particular type of living cell, or maybe a particular type of human cell. So you have a lot of layers of things that are not exactly defined in the structure that get involved in creation of a biologic. And now what are the equivalent of the generics in this field? It's essentially biosimilar, very broad definition, because it's essentially something that has same mechanism of action, so it would be an antibody if the original was an antibody. And if you pretty much use it the same way, you expect to see similar results. An interchangeable is a higher standard, biosimilar, because it would be a biosimilar that should essentially give you the same result. We're not going to talk about that right now. And very briefly on the regulatory pathway, there's when the US law was first passed, it mentioned 12 years of exclusivity, and people panicked around the world. It actually means it's 4 years of data exclusivity, and market exclusivity runs independently for 12 years, so you get 8 more years of market exclusivity. So how does this thing work? A biologic product is considered biosimilar, and then, like an ANDA, a biosimilar applicant would file a BLA, and the approval process is quite similar to a generic, small molecule generic approval process. Now, what's the problematic IP issue? As I mentioned, a biologic has to be created in a very particular way in a particular living organism, in what's called a bioreactor. Now, these generally are protected by patents to composition, methods of use, pretty standard for any drugs, but method of manufacture. And this is one case where a product by process claim, now traditionally a product by process claim in the US is allowed if the product is new. But here, every method of making it gives you a different product. So the method of manufacture becomes the key patent. And the fun part of the whole story is the FDA Orange Book does not list the method of manufacture patents. Oops. So the FDA comes up with something called a patent dance. And what the goal of the dance is, in the end, to come up with those key patents directed to method of manufacture. If you manufacture a biologic the same way as the original reference product did, a biosimilar, then you will get a product that's very close to the biologic and meets the biosimilar and functional and clinical functionality. So since the FDA does not, Orange Book, the FDA's Orange Book does not provide the method of manufacture patents, the only way you can get to the method of making patents is directly from the reference product maker. And the whole dance is designed to get that out of the reference product maker. Okay, so there's the dance, and I'm not going to go through the dance. It's initiated by the biosimilar applicant who says, I have marketing approval, and essentially asks the other side, the reference maker, to start the dance. And the first step is to say, I have marketing approval, a pre-marketing approval. I'm going to market a biosimilar, and this is how I made it. And then there's a whole lot of steps. I won't go through it, of specific time periods to exchange information, the final result being the reference maker knows how the biosimilar maker made their biosimilar, and then they tell them, well, this is our method of making patents, and we are going to assert these against you. Because I mentioned before, if you make slight difference in the method of manufacture, the actual end product of a biologic, even it meets the definition of a biosimilar, is not structurally identical. So a composition claim, the claim to a structure, is not going to meet the definition. So here's the case law that created a lot of fun and exposed some of the problems with this system. Amgen makes a product called 175 amino acid growth factor. It treats neutropenia. Neutrophil is a certain cell in your blood, goes down to very low levels in patients suffering from neutropenia, and given them GCSF, granulocyte colony stimulating factor, would increase the neutrophil levels. So Sandoz filed the first ABLA application, and then Sandoz did two funny things. First, it informed the reference product maker, which is Amgen, that we will someday market this. It was not necessary, it was not part of the patent dance regime. It kind of gratuitously did that way before it was approved. Once it was approved, it once again now said we have been approved and we will market it. But they refused to give Amgen how they made it, and therefore did not completely finish the first step of the patent dance. Now, the statute and statutory language is important. The statute uses the term shall. If we go back to and look at those multiple steps I spelled out in the earlier slide, it was A shall do this, the other person shall do it. But essentially what the Federal Circuit did, and which is what Sandoz had argued, is if a statute says you shall do something, but the statute also provides a remedy if you don't do it, then the shall becomes a should, which means you don't have to do it. It's an odd thing because now, by not doing the dance, the reference product maker, which is Amgen, can sue Sandoz, but Amgen does not know what Sandoz is doing and how they are making it. And it essentially remains up to discovery and litigation. So the whole regulatory scheme seemed to have failed right there. So Sandoz won in district court and won in the first round in the Federal Circuit. And the result made both sides unhappy. First of all, in a 2-to-1 decision, Federal Court said the patent dance is not mandatory. If it's shall and has a remedy, you don't have to do it. Then a different combination of two judges said, well, you can only give the notice, the pre-marketing notice. Pre-marketing notice starts the litigation clock. So it's important when you give the notice. You can only give after you get approval. So this Amgen was happy with, Sandoz was not. So an en banc panel appeal was filed, and Federal Circuit declined to hear it for no reason. And one would expect that this case would go to the Supreme Court on the first ground, because rarely this, I would say never, this situation has come up when a statute says shall do, and somehow the way the statute was drafted left a loophole that shall doesn't mean shall. Just a list of not everybody has gone the path of Sandoz. Some have not. And the patent dance is being danced in the US, for whatever that's worth. So, what now? What should in light of all this, um, reference biologic maker do. And this is my last slide, because this is something that has really laid open a question and kind of put the whole way we think about new drugs and new products on its head. Because now it can create a situation where the most critical patents you're asserting against somebody, you don't know if they've infringed. Firstly, can you really assert that? You don't know if they're using the same method you used or you have patented. So that's problem one. And the second situation comes up with what's really the value of the patent of method of making to the owner. All it's doing is disclosing in the specification how to make it. So essentially giving a biosimilar maker a wide open license, like this is how we do it, this is the way to do this. But looking at for the other side, if you don't apply for that patent, and that patent is not in the public domain, the biosimilar maker has to go through a lot of testing to figure out if their product works. So that's, I think, where things are standing. It might make trade secrets sexy again if somebody can keep trade secrets in this kind of in the biotechnology area long enough. And if you need more information from these slides, I went over some of them quickly, let me know and I'll be happy to email you a copy. Thank you.